I'm Dave Nichols, undergraduate teaching coordinator, Department of Animal Science, and I'll be serving as MC this morning. It's a pleasure to be here. Our students are extremely ready for fall break, and so Ernie, they're trying to get out the doors. And so anyway, it is a pleasure to be here. And my first duty is to introduce my boss, Dr. Ken Odie. Dr. Odie is head of the Department of Animal Science. Dr. Odie is a native of Pollock, South Dakota, has a ranch that borders almost the North Dakota line. He received his BS degree from South Dakota State and received his DVM and PhD here at Kansas State University. He was on the faculty at Colorado State, North Dakota State, as well as some time in the industry before coming here to Kansas State University as our department head in 2007. Dr. Odie has done a wonderful job leading a very diverse and large faculty, student numbers increasing. Please welcome Dr. Ken Odie.
Thanks, Dave, and good morning and welcome. It's always my pleasure to come over here and I have a chance to share a few comments with you at the start of Swine Day. This is actually my 9th Swine Day. I was just counting them up this morning, so I don't think I've missed any, and they've all been great, but they seem to just get a little bit better each year. I do want to just share a few comments with you, maybe a few comments about the department. So we actually are the largest department at Kansas State University. And there's a lot of ways of looking at that, but I'll just give you a few numbers here. We have over 1,200 undergraduate students, over 200 graduate students. We have 55 faculty. We have 115 full-time employees in the department. We have all the livestock units here north of town, and we feel really fortunate at Kansas State University to still have our livestock units very close to campus. And we think that's a huge advantage for particularly our teaching program, but also our research program that's able to have facilities that are close by that our faculty can use in both our teaching and research programs. And we've had a lot of upgrades to those facilities through the years, and certainly the swine unit as well, led by our swine team. So I'm just really pleased to have a chance to come here today and share a few of those thoughts with you. I do want to make a couple of other comments. About a month ago now, I attended the Swine Feed Efficiency Conference in Omaha, and I think many of you were probably up at that meeting as well, but I want to share a few thoughts about that. Number one, I thought it was an outstanding conference. Leadership from Iowa State University and our team here at Kansas State University. But just as importantly, that conference is a result of a USDA CAP grant, a federal funding for research. And in that case, a pretty sizable amount of federal money. And that money has been invested to really allow a group of people to really make what I think is some significant progress in improving feed efficiency, but perhaps just as importantly, really understanding all the nuances and complexity of feed efficiency. All of these traits are very complex, and particularly when you start selecting for a particular trait, there's a lot of things that may potentially come with that. I thought that conference was outstanding. I learned a lot going to that conference. It's well organized, but one of the points that I want to make is for us to really sustain and do well what we do at an institution like Kansas State University, we need your help, and many of you do fund work here with our swine team at Kansas State, and we appreciate that, but we need to sustain public funding in our universities as well. And I'm talking both about state funding, I'm also talking about federal funding, and having the federal funding available to do the work that we really need to be doing. Obviously, you guys also support work here through the Pork Board, and we are also appreciative of that as well. The other comment that I want to make is many of you know that the industry, the swine industry, faced a huge issue here a couple of years ago with PED virus. And today you're going to hear a little bit about some of the work here at Kansas State University in that area, but I'm very impressed with the industry's response to that, and particularly the people here at Kansas State University and Animal Science, College of Veterinary Medicine, and the role they played in helping address that particular issue. So my thank you to all the folks here and for the work they've actually done in that area. I'm not going to be able to spend much time with you here today. I have another obligation on campus, but again, Welcome to Kansas State University. It's a great time here. Football hasn't been quite so good this year, but we'll get that turned around one of these points in times. And enjoy the day and enjoy the program. Thank you, Dave Nichols.
Thanks, Ken.
Well, thank you, Dr. Odie. And through the day, I'm going to make a few introductions of some of our honored guests here. And first, Dr. Ernie Minton. Ernie, will you stand up? Dr. Minton is our, make sure I get the title right, Ernie, Associate Director for Research and Technology Transfer. So thank you for being here. As you guys have all come to expect, Swine Day is always a put together program. And to kick things off, it's my pleasure to introduce three faculty that are going to share the mic in the first presentation. Dr. Dwayne Davis, Dr. Jim Nelson, Dr. John Gonzalez. Dr. Davis is a native Kansan, and we have a lot in common in that he and I came to Kansas State, or he came back to Kansas State, I came here for my master's in January 1977, and that's when Dwayne joined our faculty. Dr. Davis completed his PhD at University of Missouri and started here in the spring of '77. One of Dr. Davis's first graduate students was Dr. Jim Nelson, and Dr. Nelson got his master's under Dwayne and then went off to Nebraska, finished his PhD in 1983, and then took the swine extension position I had when I moved into the teaching position at Burl Coke. And finally, the third presenter is Dr. John Gonzalez. John is one of our more recent hires, outstanding researcher in growth and development that comes to us from the University of Florida. To present the topic, Production Consequences of Low Birth Weight Pigs, Dr. Davis, Nelson, and Gonzalez. Please welcome these guys to the podium.
Thank you, Dave. Is this on? Can you hear me all right in the back? Well, this is an exciting day for us at K-State. It's always fun when your administrators come in right away and they start asking you for more money. My job is to thank you for the money you already gave us, okay? The sponsorship out here today, the trade show is phenomenal. The interaction, we ran around town a little bit last night, had dinner and a couple meetings, and just the interaction, fellowship, and friendship of all the people in our industry here in Kansas. You know, we're not in Iowa, we're not in Illinois, but I don't think some days we want to be. But we have a very special bondage within our friendship and family here. We welcome you all back. Also, all the people that aren't in the trade show that actually sponsored other events today. We've got Farmland and Soother Feeds, that the tremendous pork lunch you're going to have with all the trimmings. That's a sponsorship that doesn't show up anywhere except on the signs. And then also Elanco for the ice cream. Are you going to stay around for ice cream, Nichols? Yeah. We can't miss the Call Hall ice cream after the program. So we really thank all those folks and everyone for coming and presenting all their information in the trade show as well as in here. This is really our swine Thanksgiving. Everybody gets together and we have a chance to share data, but really it's the interaction with people. We leave the lunch extra long, Aaron, before your talks this afternoon, just because we want you to interact and have a chance to catch up with one another, because the world today is pretty busy, isn't it? Grad students, you know, you look all dressed up today. I usually see at the farm out there weighing pigs and collecting data and people teaching and just all that. And so it's an opportunity for us to simply say two words: thank you. The program today is outlined. I think most of you have seen a copy of that, but I want to highlight a couple things here. One is we're going to get updated on the VFD at 10:30 this morning. We have some other fun right after that we'll talk about here a little bit later in the program, but then we'll go to lunch and come back. We've got the latest update. The group's going to talk about the latest update on applied swine nutrition. Special talk, and Dr. Odie alluded to that fact a little bit ago, is the whole feed processing and PED. And then our keynote, one of our keynote speakers, and we're delighted to have you here, Aaron, is key profit drivers, because as all of you know, the reason we're able to continue to have swine industry days is because you're all involved in an industry that we hope is profitable at the end of the day. Because if it's not, with technology and advances, then we probably won't have Swine Day very long. Okay, and Dave did a great job introducing. I don't need to spend a lot of time on this, but as I put this together, I thought of team building and working together, and there's a special person today, and I don't know where he's at, but really built the K-State swine program. I don't know where Dr. Ali went. Did he take off? Okay, stand up. How special it is to have the person that built this program. Thanks for coming, Gary. What a special day for us. And we know that, you know, I remember you telling me, you know, Jim, I don't want to leave research, but I got to follow my goal to become an administrator. And I remember telling you I thought you were crazier than hell at the time. But the point of the matter is you followed your dream. You made a lot of money as an administrator, and now you're in the cattle business. What happened? What happened? Okay, so we're going to get right into the topic. It's about team building and addressing reproductive physiology, muscle biology, swine nutrition, and management. Last year I used this slide, and I probably didn't address it very well, in that in the swine industry today, and many people are addressing this in different conferences anymore, is where we're really lacking is innovation in research, in my opinion. And it's not to say that, you know, I think I misled people by last year by saying, you know, we spend so much money on marketing, we need to put more sand up in the top of this glass in innovation. And the real reason, quite frankly, that the plant science people, the agronomic industries are just smoking us is so much more money goes into that research. If you look at the money into plant research, Gary, you were an administrator, I wasn't even close to what we get. And so we need to figure out a way in animal agriculture to balance and come up with innovation, refinement, which a lot of the people will talk about that research later this afternoon, as well as a lot of the marketing research is done. The problem, if we're going to feed the world, to be honest with you, or at least be part of the solution of feeding the world, is quite frankly in the pig business, in my opinion and many others, we need more innovation and research, more discovery. And so that's what this team's about. We work in the top of the hourglass, and we spend a lot of time studying that. This is the group. Not very big, but it's a diverse group that really understands muscle biology, reproductive physiology, and we got Hauser's in there, Dr. Hauser, that's really good on the carcass cutout, evaluating a product all the way to what the consumer wants. And so this group works really hard. Our areas of emphasis are pig survival and ultimately pig value. I think you call it grade A pigs in your presentation. So we want, some people call it full value pigs, value realized pigs, as well as the survivability of those piglets. I borrowed a slide from Alan Schinkel at Purdue, and they've been collecting a lot of commercial data as well as many commercial operations have studied, and I mentioned this last year. As you continue to increase the number of pigs born alive per litter or total born alive, total pigs born, survivability and commercial production continues to drop. And Alan's data, when it goes over 16 pigs total born, and Gary talked about this at dinner last night, number of pigs weaned actually doesn't change. And so we've got a survivability issue in those production system, pre-weaning survival issues, and there's a lot of people other than just our group that's addressing that in commercial production. I want to give Dr. Schinkel credit for his efforts in that arena as well. I got a video to kind of show you. I thought I'd break up the data a little bit to show you what happens with a lot of these sows, and this is actually our farm. This little pig, I was out there, we were collecting data, weighing some birth weights, and went down and studied this little guy. This is what happens a lot with these smaller pigs in a litter. They keep trying to eat, trying to get feed, and it's kind of like, I didn't know what the right analogy was, but this is kind of what happens. This little guy is like the KU football player in about 2 weeks. He keeps getting knocked around by the bigger pigs in the litter, and you'll study him. After a while, he just quit and gave up. You know, he says, I've just had enough. And honestly, you know, a lot of people talk about these piglets dying the first 3 days. I talked to Dr. Dean Boyd the other day, and in their production system at Hanor, a lot of those pigs are dying later in the production system, much like this little KU football player in this particular scenario. Dean was kind enough to send me some of the data that they've been collecting and analyzing at Hanor with the Triumph group. And he continues to emphasize, like everybody, more pigs, more pigs, more pigs, because as you know, it dilutes the wean pig cost and increases the number of pigs marketed. Last year I got in a little trouble, and luckily my boss had another meeting today because I said these little pigs that we continue to have to figure out a home for or a place for, it was an animal welfare issue to our industry. And my boss said we're not supposed to probably elude that. Well, in Europe, and matter of fact, in the United Kingdom 2 weeks ago, Dr. Dean Boyd, not Jim Nelson, said this is going to become an animal welfare issue. And so we've got to figure out as an industry how to solve this issue, keep those pigs alive. Dean also said there's clear financial benefits in terms of it's a lot of dollars. He just didn't know how many total dollars per pig that they market in their system. He also suggested, and Hanor studying this, there's a large genetic component to all this as well. And so that was interesting. Julie Feldposh actually helped us a lot with some of our fetal development, fetal programming research here at Kansas State. And what she put together was a literature review that will be published this next year, whereby published data shows as we increase litter size, we continue to shift birth weight on this axis to the left, where the tail is not normally distributed, and the tail goes lower as we continue to increase the number of pigs born alive. And if you drop down, if you come straight down, if you look at research published data, survivability and birth weight comes right down with that as well. And I'll talk more about that in just a little bit. For some reason, that slide doesn't want to come through. There it is. The consequences, and I'm probably getting this a little bit in head, but students always ask me, what does this really mean in the real world? You know, other than Leo, we lose a few more pigs. You know, what's it mean in the real world? Well, if you track those pigs to market, the light birth weight pigs, those that weigh about 2 to 2.4 pounds, versus the heaviers, 3.9 to 4.4, you pick your market weight, it's between 12 and 14 extra days, slower time that those little pigs take in the production system. They simply are a cost all the way through. It's just not in the farrowing house, but they're actually a cost all the way through grow finish. And the other challenge is if you keep gilts back out of those, smaller pigs have lower litter size, and they also then transcend or have lighter birth weight piglets as well. So it's an evolutionary thing that just continues in production. The only other slide that I borrowed from Dean, and the reason I borrowed this one from him is Mike Johnston was here. Yeah, he may still be outside. Mike, where are you at? Right here. Okay. When I was talking to Dean on Tuesday, he goes, Jim, we've done a lot of analysis of birth weight and following those pigs' weaning weight and all the way to market. He goes, a K-Stater actually weighed all those pigs. I go, well, who might that be? And he goes, well, my name's on the slide, but it's Mike Johnston. Thanks for weighing all those pigs for Dean. But anyway, if you look at this slide, and it's actually the Cabrera data that's recently published, shows, and I just want you to drop to the bottom line, those little pigs also have lighter weaning weights. And if you make the comparison here, a pig that's weaned at 7 pounds, lighter birth weight, lighter weaning weight, versus 12 pounds, their eventual market weight in a fixed time system or wean to finish, in Dean and Mike's data, I should say, 223 pounds at the same time versus 289. And Dwayne was on the conference call, and I said to Dean, I said, my God, if you came up with a product or a technology, Wayne, that got you 70 pounds more pig, I think every producer in the U.S. or the world would buy it and probably not even ask the price of it, right? Not even ask the price. And Dean says, well, we would ask the price, Jim. You got to understand who we are. And I said, I do get that part. Bergstrom, John Bergstrom, is he here? All right, John? John really led up a very comprehensive study for us several years ago where we followed pigs, and again, this experiment was designed to actually study something slightly different, but we took birth weights of pigs and looked at, as they approached 220 pounds or greater, how many full value, or you call them grade A pigs, that we actually realized. Well, look what effect birth weight has on the percentage of pigs that reach full value. There are differences between gilts and barrels. Gilts grow slower, we all know that. But the point is, if you come back and get into these 3.5 pound pigs, you'll get 80 to 90% full value pigs. You come on down, get these 2 to 2.25 pound pigs, you're in that 40 to 50% of those pigs actually reaching full value or grade A pigs in the mash-off production system. So it's huge, and we did a comprehensive study then and had several cooperators trying to understand where the cutoff is for these pigs that what we called were at risk. And John, we used your data exclusively in some of the modeling efforts that we used. And so our goal has been to identify what we call are these at-risk pigs, the pigs that are going to give us trouble, or actually be those pigs that either fall behind in farrowing or don't perform well in the finishing. And we had a lot of help with this. And I can't get into the specific details, but we had several statisticians working on this project. And we use birth weight as our threshold indicator for survivability in this case. Trying to understand in the farrowing house, which pigs were going to have mortality issues. We did a meta-analysis. We combined data. These are individual pig weights. And Dale Keysucker was here. One of the farms we worked with was Dale and Keysucker Farms. John and I think Megan was in on that. Several people helped weigh all these pigs. Well, the Europeans were doing the same thing, taking a lot of birth weight information. And so there were 4 different farms used. We said 2 studies, Dr. Henry, because one was Europe, one was the US. So the statistician said those are 2 separate studies. And so we compiled 4,068 observations of birth weight and pre-weaning survivability. And we appreciate all the work that went into that. We used a mixed effects logistic regression model. If you got any questions about that, talk to Dr. Dreech later. Okay, so this is what we did. We actually tried to discover where the inflection point changed, and this is under commercial production systems. Where's the pigs, Aaron, we called them at risk. We didn't know what else to call them. And if you start following back, and I apologize, this is in kilograms, so start thinking about pigs that weigh 4 pounds, 3 pounds, 3.5, and as you get back to 2.45 birth weight, the inflection point changes. And suddenly, the ability of that pig to cope in modern swine production drastically changes. From there, and quite frankly, their chances of surviving change dramatically as well. And so with this data, the 2 leading scientists here started discovering what's different about these pigs, and they're going to talk about that a little bit later. I want to show you a couple more slides because people say, well, what % of the pigs, Gary, are we talking about? Is it half a pig a litter? Is it 1 pig per litter? Well, it's actually 20% of the pigs in this study. Of the 4,000, 4,100 we studied, it's 800, no, it's 800 to 1,000 pigs out of the production system. Dean's always trying to figure out separate flows for those pigs, how to manage those pigs different. The point is, it's pretty significant. And actually, I didn't put in another slide that Dean showed in the UK, is in 2003, the number of pigs in the Hanover system that were at-risk pigs he did it on a litter size basis, was 0.7. And in 2014, it was up to 2.4. And so this issue continues to increase as we increase litter size, and most of us already knew that. Okay, so conclusions. Birth weight is our indicator. It's what we're going to use as our threshold indicator. Is it the only thing? These scientists can argue that we should be doing many different issues, and we'll let them talk about that. But we think in production, that's the best measure we can take under commercial production system. Having said that, the cutoff seems to be about 2.45 pounds. And so that's where people are thinking about, okay, we need to work differently here in terms of prenatal. We call it fetal programming. Different people call it different things, but the whole fetal programming, or as Dr. Davis will talk about a little bit, also the opportunity there for intervention under production systems, and those are many. But we believe that successful intervention will take on different strategies. We think there'll be a prenatal component. We think there'll be a huge opportunity in gestation. Dr. Davis talks about epigenetics. It may be multi-generational. It may carry on for several. But as we got the muscle biologists, we think there's huge opportunities in the whole muscle skeletal development of these piglets. However, if you look at the overall approach, if we start going into the whole prenatal area, we hope that that eventually results in that increased growth rate weighing out into growth finish, that we get those pigs that are lagging behind 70 pounds, that we can pick some of that growth up. And in the end, and you're going to talk about this, we hope it influences profitability, right? Because that's why you're here, and that's the drivers that you'll talk about. Okay? At this point, I'll turn it over to Dr. Gonzalez, and thank you very much. Thank you.
Good morning. The role of my lab over the past 2 years in this group has been to explain what the muscle differences are between these large, medium, and small birth weight pigs. And when you think about what determines the ultimate muscle mass of an animal, there are 2 major determinants. First being the number of muscle cells that have been formed in that animal, and the second being how big those cells get all the way up into market. Now the one, I guess you can call it a bad fact about muscle development in general in mammals, is that the number of muscle cells are determined prior to birth. And so that makes gestation a very important time period in an animal's life if we're talking about making an animal very muscle heavy. So if we actually concentrate on the events of fetal muscle development, there are two primary time points that are associated with muscle fiber development. The first is primary myogenesis, or muscle fiber development, which occurs from around day 25 to day 50 of gestation. During that time period, animals form what are called primary muscle fibers. These are denoted up here in my photomicrograph as these orange donut-shaped structures. Now, the role of these structures are to serve as a scaffolding for the second wave of myogenesis, which occurs from day 50 to day 90 of gestation. And so if we actually take a histological picture of these animals at day 90 of gestation, it ends up looking like this, where we once again have our primary muscle fibers, or each of these orange dots. They're surrounded by secondary muscle fibers, which are the red circles. Now, through our research, we've estimated that there's anywhere between 22 to 27 secondary muscle fibers that are formed per primary muscle fiber. After that time point, the major developmental event that occurs is you get enlargement of these muscle fibers that are formed. Also, I've included in this graph, and this will lead into part of Dwayne's talk, we do have adipogenesis competing for nutrients, or the formation of fat cells. And I'll leave that to Dwayne because he likes to talk about fat more than I do. I'm a pure muscle guy, I guess. So last year we presented data where we actually took 36 sows and we harvested them on day 60 of gestation. We took out the small, the medium, and the large body weight, our pig, and we determined that based off of the crown to rump length, or the length from the head to the tail of the animal. And we looked at 4 attributes of interest as far as muscling was concerned. The first being whole muscle area, or the size of the loin muscle, and we concentrated mainly on the loin muscle because I guess that's my meat scientist, where we always want to go back to the loin muscle. We also looked at the number of primary fibers within that loin muscle, the size of those primary fibers, and then at that time point, because you're at day 60 of gestation, so that's the transition point between primary and secondary myogenesis, we physically counted the number of secondary fibers that were present at that time point. And for the balance of this presentation, what I'm going to do is basically pick on the small fetus and always compare the small fetus to the medium and the large fetus individually. So when we actually ran the statistics, we ended up finding that there were significant size effects for the whole muscle area of the loin, the number of primary muscle fibers formed, and then the number of secondary muscle fibers formed at that point in gestation. So if we compare the medium to the small pig, the medium fetus had 13% larger loin muscles, and this was due to a 12% increase in the number of primary muscle fibers that were formed at that time point. Now if you compare the small fetus to the large fetus had 21% larger loin muscles. This was due to a 13% increase in primary fiber number, but also an increase in the number of secondary fibers that had been formed at that time point in gestation, which is pretty neat. So for this year's data, we actually decided to push out gestation to day 90 and see what developmental differences there were between those 3 classes of fetuses. And what we ended up finding out, let me just set up my slides, and they'll all be the same throughout the rest of my portion of the presentation. I'll always have the size of the fetus along the x-axis, where I present small first, medium second, large third. And then I'll always have my attribute of interest on the y-axis. And in this case, we're looking at the overall size of the loin muscle once again. And so what we ended up finding out was there were size differences similar to what we found in the day 60 study, with medium pigs having 20% larger loin muscles at this point in development than the small pigs, and the large fetuses having 30% larger loin muscles than the small fetuses too. So the cool thing about this, if you actually think about it, and we think back to that previous slide, the developmental differences have actually gotten worse as we've advanced in gestation. So we've gone from, like, if we use the large, a 21% difference to now a 30% difference. So things are getting worse for the small pig. They're starting to lag behind these other 2 types of fetuses. So what we ended up doing is looking at primary fiber number, once again counting the number of primary fibers that are present in the muscle. We actually at this point do not have a size difference anymore, which we did have at the day 60 part of our trial. And so everybody has the same amount of primary fibers, which actually started getting us to think, well, maybe the small fetuses are just at a delayed trajectory of muscle growth. And so at this point, they've finally caught up with primary fiber development and have the same amount of primary fibers as the medium and the large pigs. We looked at primary fiber area, the size of those primary fibers. Similar to the last study, there was no size effect, and all treatment groups or all size categories had around 150 square microns in size for their primary fibers. We now go to count our secondary fibers that are formed, so we're right at the end of secondary fiber formation. We were expecting to have differences between our medium and our large pigs, and you can argue, statisticians can argue if I ran a linear contrast that would probably come out as significant, but we actually compare the size differences. There is no size effect at this point, which led us to kind of start to put together the story that once again the smalls are lagging behind in muscle development from the medium and larges. And at this point in gestation, visually, yes, they look like they're still behind, but they're starting to catch up as far as the number of muscle fibers formed. When we look at fiber area, our size effect reappears, where our medium pigs have 13% larger secondary fibers than our small pigs, and our large pigs have 17% larger secondary fibers than our small pigs. And so at the end of the day, if we go back to our normal development graph of myogenesis, once again to remind everybody, normal development would go from day 25 to day 50 for primary myogenesis. Secondary myogenesis is going to take over after that, go to day 90. What we hypothesize is going on with these small pigs is everything's getting shifted to the right and getting delayed. So they're having delayed development, where you end up having a shift in primary myogenesis. This causes a shift in secondary myogenesis. In turn, if we're going to just concentrate on how much muscle the animal has at birth at 114 days of gestation, there's less time for the muscle fibers to hypertrophy, and so that's going to cause these smaller pigs to come out with smaller muscles in general. And so our overall conclusion at this point from these data is that secondary fiber hypertrophy is responsible for the whole muscle differences when you compare a small fetus to a medium and a large. Now, one last thing I wanted to present, and actually I told my PhD student, Matt Vaughn, I would give him credit for this because he went out and actually sought out this data on his own, and it actually tells a very good story as far as postnatal development. And he went into our photos and counted the number of satellite cells that were available. So those that aren't familiar with what a satellite cell is, these are muscle stem cells that are responsible for all the postnatal muscle growth that an animal will experience. And so these guys lie on the outside of a muscle fiber, and as we need more protein synthesis, these guys will become activated, incorporate in, and contribute to postnatal muscle growth. And so we identify them through the protein PAX7, and when we ended up counting them and running the statistics, we once again have a size effect where medium pigs have 13% more satellite cells than small fetuses, and then large fetuses have 24% more satellite cells than small fetuses. And so, the take-home message on this, if we think about, like Jim just talked about, that these guys have slower growth rate, they're not going to be as big muscle-wise as their medium and larges. This is probably one of the reasons why. They just they're not physically born with the machinery and in the terms of satellite cells to have them grow just as efficiently as a medium and a large pig. And with that, I'll let Dwayne take over and follow us through.
Thanks, John. I actually, since you started, I've been wondering if that comment about me being the one with the fat was a slam or what. No. But I figured it wasn't because you were taking credit for muscle, and I didn't see that. Okay, so to sum up what Jim and John have been talking about here, prenatal growth restriction during pregnancy, we know that's where it's starting. It's probably got to do with the amount of support that the placenta is providing to these individual fetuses. Clearly in pigs it doesn't get divided up equally. That changes fetal development, low birth weight. We kind of all know this, and there's data to support it, that does reduce their colostrum intake. As Jim indicated, clearly that increases death losses, particularly really early, and maybe some of them delayed a little bit later in lactation and Dean Boyd's data. And the big bottom line is there are fewer pigs and fewer full value pigs available. So that's a pretty big deal, and it's a pretty big deal to the whole industry. John was concentrating on the muscle data that was collected from these studies he was commenting on. Again, these were at 60 and 90 to 95 days when we were collecting these fetuses. These aren't the fetuses, these are some examples, of course, of pigs at birth. And the top one is a pretty nice looking pig. Bottom one has some problems, clearly. And you can see that throughout that pig's body. If you just looked from the shoulders back, you know, it just wasn't filled out and plump. See some ribs, some other things. Up here in the head, and this is something the Belgium researchers have pointed out, there's a big difference in head shape. And in fact, besides birth weight, you can probably just look at the head and pick up a lot of these early things of intrauterine growth retardation, where a bigger pig has got a pretty flat slope to the forehead. This one is pretty rounded. In fact, they refer to them as having a porpoise-shaped head. And so that's what's going on. Now, as far as the pig itself, all the systems in that pig's body are being affected by being deficient in its nutrient availability. But the one that it really tries to protect, the highest priority, is the brain. And so we say that they do brain spare or brain sparing. And so classically, this is a way to look at it. You know, you can take some measurements to predict that, or you can actually, as we did, remove the brain and the livers, what we used as to create a ratio. So the brain gets the priority. Really, all the other organs come in after that, but the liver is a pretty good representative one. And so if you take the weight of the brain in those fetuses and the weight of the liver and create the ratio by dividing the brain weight by the liver weight, by 60 days, that is actually a significant difference between each of these size fetuses, but it really gets magnified by 95 days. And that's the point that John was pointing out on the muscle. You know, the muscle is also in the same situation as the liver. It's taking a back seat because the pig's doing everything it can to keep the brain developing as much as it can. And I think that's not a unique finding for us. We do repeat what other people have shown. I think an important point is that it's just not the smallest ones that are affected. Those in the medium part of the litter, as far as size, are also. And granted, our focus right now is on these smallest pigs, but I would suggest to you that there's a lot of pigs in there that are being affected, and someday we may turn our attention down here and try to improve the rest of those to even make a fuller value pig at market. So this is Dr. Gonzalez's slide. He said I would talk about fat. So adipogenesis does start back here in the pigs before mid-gestation. It really, at this point, what's happening are preadipocytes. The fat cells are adipocytes, and the preadipocytes are dividing. Okay, that's what happened initially, and that's happening throughout most pregnancy, right here. Now, if you go back to these two pigs I used as examples, and I'm calling the smaller one damaged because I think it is damaged at this point in all of its organ systems, actually. This is one kind of generalized way to look at adipogenesis. So again, it increases, but by the time, And Dr. Gonzalez worked on these for me. He got gestation shifted over a little bit, but that's okay. So back here, around 112 days or so, the adipogenesis, in terms of proliferation of those pre-adipocytes, starts to shut down. What's really happening is they're differentiating to adipocytes so they can go ahead and fill with fat up to that point. Now, you know, baby pigs don't have hardly any external fat. Okay, that's why they've got to rely on the glycogen in their liver and some in their muscle, and the colostrum's really important to them for that. What happens in the smaller pigs is they're doing just the same thing that John referred to in muscle. They're shifting that to the right. So those cells continue to proliferate, and actually they continue to proliferate here into the postnatal period. So if we shift those numbers back that way, and probably it's not until about a week postpartum or after they are born before they really get to the same point that the bigger pigs are before the time they're born. What that means is there's longer time for them, for those pre-adipocytes to keep dividing. Okay? So they keep dividing. That means even though the little pig has got less fat at birth, eventually it winds up with more because pre-adipocytes have longer to divide. You wind up with more of them, so when they start to fill with fat, that's why these little pigs are probably, I think, the main reason these little pigs are fatter. These are some data from France that explain partly why this happens. And this paper has a lot of data in it, but again, I got scrunched over here. But this is the expression of a particular gene, DLK1. It's actually a classification that's considered immediate early in a lot of different systems. But the important thing in adipocytes is that it needs to remain high for those pre-adipocytes to remain proliferative, for them to remain pre-adipocytes and keep proliferating. And what's really happening is here at 71 days of pregnancy, the median weight pigs in their study had a lot higher expression of that than the small pigs. Okay, so that's maybe okay. We don't know too much about what's going on there, but that's probably keeping both of those pigs generating more eventual fat cells. But by the time you get to 112 days here, the median weight pigs have decreased expression of that gene. So their pre-adipocytes are differentiating to adipocytes. They're going to quit dividing. But the little pigs still have pretty high DLK1. And out here that was actually 2 days after birth, they still have a lot higher. So that higher expression is an indication that the regulation of the division of those cells is continuing such that they can divide. And then again, they're going to wind up with more adipocytes. So this is kind of a challenge here. Besides the muscle side of the story, increased fatness, decreased carcass value. Now, colostrum, well, kind of know small pigs get less colostrum, okay? So Jim says this is Thanksgiving. It's not for me. Next week's Thanksgiving, but this is a great day. But so next week, we're probably all gonna, or at least I always eat more than I need on Thanksgiving, right? And fortunately, there's people that take care of that situation always. Maybe some of you do that, so that there's more than enough so that people that are smaller or bigger or whatever all get all they want, plus probably a little bit extra. Now, it doesn't work that way for the relationship between the sow and the litter, because the bigger pigs, and maybe the ones that are born earlier, we need to look at all that a little bit closer, I think, get more of it. So this one rule of life is those that have get, and that just definitely happens in these litters. There's different ways to try to estimate colostrum production. These are data from Belgium. I took some of their numbers and put it into pounds, but for example, you can calculate that a 3-pound pig probably needs about 36 grams more colostrum than a 2.5 pound. That's what he needs, but on average, he's probably going to get almost twice that much. So again, that doesn't just leave as much for his smaller contemporaries. These are some really recent data from USDA. Well, I say recent. This paper was actually published in 2015, and USDA at Clay Center in Nebraska. Dr. Jeff Valle has led this research. This was data based on about, in this case, the paper says over 15,000 pigs, and they generated the regression lines. Now, what this is measuring is what they have called immunocrit, which is a different way to measure immunoglobulins in the blood of the little pig. So it's possible by several methods to measure how much antibodies actually have crossed into the little pig when it nursed during that first 24 hours. What they've done is developed a lot simpler method to do it. So basically they take a blood sample, they spin it down, separate the serum, and then there's a precipitation step that just precipitates the immunoglobulins. They spin it in a little centrifuge and measure the height of that column. So this lets them measure it on lots and lots of pigs fairly quickly. And what they've done here is taken their data, generated a regression line which showed an important effect of the amount of immunoglobulins in the first 24 hours, or blood taken at 24 hours. And then here they're plotting lines for the pigs that got the least colostrum and the most to illustrate what that probably means. First thing is that weights at all of these time points do show that the pigs that got more immunoglobulin in the first 24 hours, probably they got more total colostrum, actually way more. Okay, so they've taken this out to 200 days, and you know, this may be the clearest picture we've ever had of how much effect colostrum consumption, measured in this case as antibodies, I'm going to assume that's what it's really measuring, is colostrum intake, is how much effect that has on growth to 200 days. So besides that, they looked at reproduction on some of the females that came from these litters that had different measurements of immunocrit. And more immunoglobulins, these females were as much as 7 or 8 days earlier at puberty on the extremes. And when they were inseminated, produced litters, more immunocrit meant they had larger litters and increased pre-weaning growth in those litters. So there's the reproductive system clearly is coming into this whole thing. So I would say that there's interrelated indicators. We can use birth weight. We could probably use head shape, as some of the European researchers have used. If we could use brain to liver weight ratio, colostrum consumed, blood immunoglobulins. These are all indicators of what's already happened, and they're carrying through to have major effects on really all the pig systems, and that's affecting those pigs' abilities to perform for us in various ways. So I would say these are damaged pigs. Is it correctable? To be completely correctable, we're probably going to have something in gestation operating. But adaptable, the swine industry is really ingenious, coming up with solutions. We'll hear later today, I think, from Aaron about how maybe they handle some of these things a little bit. Maybe not. I don't want to set him up if he didn't put that in his talk. But I know they work on this. So it's a problem, but you know, if you flip that, it's an opportunity, because there's a lot of opportunity to gain more from these animals that make up, as Jim showed, just 20% of the litters. So that's where we're at. Hopefully by next year we'll be able to tell you a little bit more about how we might do some things about this. So, we have time for. Dr. Nichols says he's going to answer any questions you have at this time.
If you want somebody to talk about fat, I'm on.
Glad you're up here.
Is there any questions? Yeah, we do have time for them.
That's good.
Thank you. Give a good hand. Thank you.